Great. Well, then with that, I will introduce our speaker for today. I am really excited and appreciative that Susan Davies is joining us to give this webinar today. Many of you may be familiar with Susan's name and her work, even if you haven't met her. Susan is now retired, but spent the state in Maine, working for the state of Maine. She was the Biocriteria program manager, and toward the end of her tenure there, more broadly, the water quality standards coordinator and 305B coordinator. And Susan has done extensive work on the issues of biological assessment and biocriteria in terms of the technical kind of background on these subjects, as well as the practical. How do you actually make these tools work when you're working within a state agency and really trying to apply these ideas on the ground? So we're just really excited to have Susan Davies here today to share with you kind of, again, an intermediate level webinar focused on these two critical tools if we think about taking the power of the Clean Water Act and really applying it in this biological context. So Susan, with that, I will turn the next essentially hour and a half over to you, except for moderating your questions.
All right. Well, thank you very much for that generous introduction, Merritt. And this is a terrific opportunity for me. I have, over the course of my career, felt passionately about biological assessment and its importance in the national scene in trying to sustain and improve water quality throughout the country, and my experience in my own home state of Maine has really convinced me that it can and should be done everywhere in a really strong and credible way. So I'm going to start out with a fairly simplistic overview of what biological assessment is. I presume that most of you already have exposure to the basics of it, but I'll provide opportunities throughout the presentation for a little more technical detail if that's of interest to people. And I strongly encourage you to participate by asking questions, or there's also, I'll point out something that this feature that Merritt pointed out to me. Up on the black bar at the very top of the screen is a little guy with his hand raised, and there's ways that you can interact a bit by using those buttons, and then something will appear next to your name. So I really love the experience of face-to-face contact in making presentations of this kind, but a webinar is a great fallback when people are pressed for time and can't travel. So this is a great opportunity. So bioassessment basics. This slide is just to remind everyone that it's what's living in our water bodies that really is of critical importance for management. The slide, let's see if I can use this person here was my commissioner at the Department of Environmental Protection, and we got her out on the Kennebec River at one point, and then just students here learning about aquatic life. It's an important and compelling aspect of our natural resources, needless to say. Much of my presentation will be framed around a management paradigm that's depicted on this graphic, where information from the resource is collected. It's processed through all of Clean Water Act programs, and then the agency or the institution that is doing the monitoring, we would advocate should ask the question: have we actually protected our highest quality waters? Have we achieved the highest use that's achievable? Have we improved degraded waters? and when we've managed to make improvements, have those improvements also been protected? And so then the question goes back to answer those questions through monitoring. So I'll be coming back to that theme a couple of times throughout the presentation. So we need a water management framework that targets actions to achieve environmental results. This is just a basic mindset in bioassessment that we're actually looking at the resource itself. We're not looking at the quality of water coming out of the pipes, but we're looking at what the end result is in the environment. The management framework should foster setting ecologically sound goals, not just chemical quality goals. And then it should also measure and communicate what's been accomplished. Bioassessment really does provide tools to manage in this type of a framework. There's a fair amount, even with the deeply initiated, there's a fair amount of variety in how the terms biological monitoring and biological assessment are used. They're frequently used interchangeably. A little bit more of a refinement would point to biological monitoring being focused on the field aspects, field collections of the assemblage information like fish and macroinvertebrates and algae and periphyton, where biological assessment in the lingo of the Clean Water Act and the 305B reporting and so forth is really looking at what the condition is in relation to a known or established goal condition for the resource or for the aquatic life. So the assessment is kind of the determination of where it is in relation to a goal, and monitoring is all of the technical work to collect the information that will answer that question. Biological criteria are they're benchmarks that are established through intensive data analysis that become legally binding in a way of expressing what the water quality standard goals are for the aquatic life that are being assessed. Now, basically, the biological assessment takes its starting point from naturally derived expectations for how streams work, how ecological systems, aquatic systems are structured in order to maximize their use of resources, of food resources such as detrital material coming into the stream. Anyway, this is the point is not to go into great detail with this, but just to say that the science behind biological assessment is based on knowledge and understanding of the naturally derived ecology, and then we see departures from that in the face of disturbances in chemical aspects of water quality with toxic substances and physical changes in the habitat and flow regimes. So the starting point is expected assemblage makeup depending on the river size, and then changes from that expectation is really what the science of bioassessment is all about. So very simply, if you look at a natural system, this is not probably a pristine water body, but it's a high-quality water body, has certain expectations for the types of organisms that you would find there. And here's just some examples, slimy sculpin, and there's salmonids, and this would be typical of a northern temperate forested river or stream as we have in Maine. However, as we all know, these kinds of systems are subjected to a great deal of human-caused disturbance, and so biological assessment is looking at the effects on that expected assemblage makeup relative to the disturbances that are there. So again, minimally disturbed forested watershed in Maine might have something like mayflies and stoneflies and slimy sculpins. Urban development causes a great deal of physical and chemical disturbance. This slide depicts, the color scheme is a little bit difficult to see, but the yellow here are urban streams. This is Jepson Brook in Maine, so that's that stream. There's another stream over here, Hart Brook in yellow, and then over here in Stetson Brook. And the thing to note is that the greens in the Jepson Brook stream are quite reduced relative to the greens over here in Stetson Brook. And the results in terms of the numeric biological criteria that Maine has established is that this stream is attaining our highest water quality standards because it still has a relatively intact watershed with a lot of natural forest and grassland cover. This one is becoming more impacted by development with less, but it's attaining a Class C, and Jepson Brook is in non-attainment of the minimum standards. The next graphic is the same location but showing impervious surface with parking lots and roads and so forth in black here. So it's just obviously that's a major source of the biological problems that we're observing throughout the country now. It has to do with land use alteration and impervious surface. Agricultural activities, another major stressor, causing things like elevation of nutrients and many physical changes that go along with that, as well as the chemical changes from lowering of dissolved oxygen. So the result, anyway, is very simplistically is the loss of these sensitive organisms that we would have expected to occur in an intact watershed. And again, just some examples of the sorts of replacement organisms that would come in. This is a image on the top left of a stream draining a shopping mall in Maine, and you can see the algal problems there. But a little bit closer look at what biological criteria are, because many states have narrative statements about what their aquatic life goals are. To take it a step further, a certain number of states also have numeric standards or ways of assessing things. But to actually claim that you have biological criteria, it needs to be in some kind of a policy framework that's actually legally binding. Goals for aquatic life stated as quantitative benchmarks. So it takes a good deal of intensive data analysis by assemblage type. The best programs use multiple assemblages, as illustrated here, fish, macroinvertebrates, and algae or periphyton or diatoms. Many programs center their focus on just one assemblage type. Macroinvertebrates is a very probably the most widely used, or a combination of macroinvertebrates and fish. And then these determine whether or not the stated goals in water quality standards have been attained. And as I mentioned earlier, the way that biocriteria, that these benchmarks or thresholds or levels or boundaries between different condition classes that represent biocriteria, the way that they are developed is to compare location and reference condition, or an assemblage that is the best assemblage that you can find to validly compare it to. It's a very important concept that I'll say a little bit more about later on in the presentation. This, again, it's kind of an icon or a schematic of various types of information that can be typically evaluated in biological assessment. Each one of the curves here represents, kind of one way of looking at an assemblage, an aspect of an assemblage. In relation to, this would be basically the high to low biological condition or high to low value of a metric as it pertains to biological quality, and then the x-axis here is increasing stress or disturbance or effects of human activity. So basically native taxa, would decline across that stressor gradient. Intolerant taxa would decline across the stressor gradient, but things like tolerant taxa would tend to increase because they're opportunist and they are able to move in when other organisms have been lost as a result of the disturbance. Fish lesions in fish may not show up until very high levels of stress or toxic influence. Now I have just a couple of other slides here. These are mostly just to illustrate that the science of biological assessment has advanced, I think, actually well beyond the place where many state programs and EPA have taken it. It's really there's a great deal of very strong science out there about how to measure just how to apply biological assessments. This is a very large-scale study here that was put together, a summary of many studies of a whole combined index, the fish index of biological integrity. So up here is always going to be high, and it's always going to go this way as things decline. And this is looking at various studies throughout the country and higher and higher proportion of the catchment in agriculture and those kind of universal declines of fish biological condition. And another illustration just showing some presentations of data. These organisms here, you probably have heard of the EPTs, the mayflies, stoneflies, and caddisflies, or and this is in relation. It looks like this axis has been. It's not, at least on my image, it's not clear what's being shown on this axis. But the first box plot here is an agricultural effect, so that there's a certain distribution that is of the EPTs that's shown for a collection of all the sites from agricultural lands and then forest lands, and then kind of a decline in the EPTs with a mix of residential and natural, and then the urban impacts are the greatest, illustrated by having the lowest EPT scores. So it's just a way to see that the quantitative analysis of this information can be presented in a multitude of ways. Getting a little bit more complicated here. This is a 3D presentation with 9 variables that are all collected in this first stage model. This is a multivariate analysis, and the sites in red here are the lowest class of biological condition that's allowed in the state of Maine, and it's just showing the data analysis that would separate out attainment of the highest class from the lowest class. To get into the policy a little bit more,
Susan, we were going to pause here
yes.
for questions, if that's okay. We do have one, and I have one.
Oh, great.
Is it okay if I?
Absolutely.
Okay. Well, so folks, if you have questions, this would be a good time to type them into the box, or if you're on the phone line, you can hit *6 to unmute and ask your question. Well, people are thinking, I will share one that came in from Mary. She asked, which states have numeric criteria? I gave her a flippant answer of relatively few, but Susan, I didn't know if you wanted to expand on that.
Yeah, well, that's a great question. I'm glad you asked it. There, at the end of the presentation, and presumably these slides you'll have access to afterwards, so you can check some of the fine points. But there is a web link up on the EPA website that provides a rundown of all 50 states and where they stand in terms of biocriteria development. Numeric biocriteria has a variety of there's quite a range of technical sophistication in terms of use of numeric biocriteria. Years ago, when Maine was just beginning to develop our model, there were some states that had numeric biological criteria, but it was just one variable. There was an example state that had just one species diversity index, and that was the entirety of their numeric biocriteria. Other states have done much more advanced and sophisticated analyses over the years. But probably the best thing would be to check that EPA website. The document is dated 2002, but it's my opinion that there's been, since that time, there really has not been, EPA has not made biocriteria a priority, and consequently, many of the states also have not felt that pressure from EPA. So things haven't changed very much since 2002. But I would say this is just a wild guess, there may be, maybe 5 or 6 states that have pretty well developed numeric biocriteria that can actually play a role in action forcing. And that's just a guess.
That would kind of be my guess too, actually, Susan. So.
Okay.
We have one other question here also from Mary. She says, if nutrients and algae are a big problem in your watershed, how important is it to include algae indicators in your biomonitoring program as opposed to just fish and macroinvertebrates? What would you measure?
Yeah, that's another great question. We in Maine, that's where most of my experience comes from, have found we've embarked in the last 7 or 8 years on a pretty ambitious algal biocriteria program to complement our macroinvertebrate program, and it is for the purpose of management of nutrients as well as other sorts of things. But nutrients really are the big thing that a diatom or algae program can contribute to, and it's tremendously helpful there, either to look at it, by biomass or just how much algae, is produced that has a effect on aesthetics of a water body. Also, the species composition, the types of algae have just the same sort of range of sensitivities or the same type of information content as fish and macroinvertebrates do. So it's tremendously helpful. It does take a very serious commitment, and it takes a lot of expertise. So that's it takes a big investment in order to develop credible algal indicators, as it does for fish or macroinvertebrates as well.
Okay. Does anybody on the phone have any questions before we move on? All right. Well, then with that, if you're on the phone, please do hit *6 to mute yourself so that we're not hearing any background noise. And Susan, Mary says thanks for your answers to her questions. Okay. With that, we'll move on to your policy basics section.
All right. Well, the starting point is always the Clean Water Act. I always like to put a plug in for Edmund Muskie for the biointegrity objective in the Clean Water Act, because he was a real spark plug in getting that in there. He's from my home state of Maine. But basically, it is, I think, useful at the 30,000-foot level to remember that the Clean Water Act, the framers of the Clean Water Act they had multiple things that they were trying to accomplish. So, the three major things were human health and public safety, ecological health, and this would be the biointegrity objective, and then ecosystem services like economic uses of waterways as conveyances, although they put in a lot of limitations saying that couldn't be its primary use, but human enjoyment, aesthetics, and recreation. So there were a number of things going on, and it can be quite helpful to keep that in mind when thinking about these concepts like designated uses, because some of the designated uses, they're all directed to different aspects of these three major intentions of what the Clean Water Act was supposed to do. Excuse me. So designated uses are a very important concept, and hopefully, it looks like this slide has gotten a little bit out of whack too, but not too badly. These are some examples, typical examples of designated uses. So a water supply use would be directed to human health, and it would be assessed looking at ambient water quality criteria and amounts of bacteria, viruses, whatever, human health kinds of impacts. So bacteria assessments are primarily concerned with human health. Aquatic life uses, of course, are concerned with ecosystem health or aquatic life health and condition, and would be assessed by things like not only the biological response as measured through a bioassessment-based, go out in the stream and look at the organisms, but also things like ambient water quality criteria that would be set. They would be set at levels that would be protective of aquatic life, through all the various toxics research and so forth that's been done over the decades. And then also physical and chemical means of assessment can have a bearing, or physical can have a bearing on assessment of an aquatic life use, just what the flow needs are and so forth. Aspects of fishing, looking at fish tissue contamination that might be assessing human health impacts. Other aspects of fish tissue contamination that might be looking at, well, okay, how's the fish itself doing versus how's the person who's eating it doing. And then ecosystem services would be the recreational aspects of that it's a human, desire to be able to catch fish. So anyway, that's I won't go through the rest of these in detail, but we're mostly going to be focusing on the aquatic life designated use and all of the specific details that pertain to looking at the bioassessment-based aspects of assessing attainment of that use. So looking at the organisms in the stream itself. We've always argued in Maine and felt strongly that the investment in looking at the aquatic life, it really very importantly serves assessment of all of the other uses, with some exceptions, but aquatic life in many cases really is the most sensitive use, and it really, we have found that the enormous information content in a sample of aquatic life can pertain to many, many other extremely important aspects of water quality management. So it's just a tremendously important and essential aspect of assessing how we're doing in terms of managing water quality throughout the country. So you've all probably heard this over and over, but the Declaration of Goals and Policy to restore and maintain the chemical, physical, and biological integrity of the nation's waters, of course, we've done a great deal of work and enormous investments on assessing the chemical water quality and having chemical water quality criteria. Less so with biological, and it could be argued even less so with physical. So we're not really quite there yet. Again, back a little more detail to designated uses. The use of a water body is the most fundamental articulation of its role in the aquatic and human environments. This is just kind of the paradigm behind the Clean Water Act, water quality standards concepts, and all water quality protections established by the Clean Water Act follow from the water's designated use. So in red here, then, if you get the designated use wrong, then everything else, there's a really serious fallout for management, how you can actually use the forcing power of water quality standards if the designated use is wrong. And this becomes particularly important at least from my experience and perspective in biological assessment and biocriteria in terms of the aquatic life uses, because in many cases they're not very well refined or very descriptive. So again, the aquatic life use is a required provision in water quality standards designed to protect and restore aquatic life. It can, as I mentioned on that earlier slide, it can be assessed looking at the chemical condition, the physical condition, or what we're focusing on, the bioassessment-based assessment of attainment of aquatic life use. Just a few out of an enormous variety of examples of how states have thought about aquatic life use. These are just, a few little sort of commonly encountered bits of terminology that often appear in water quality standards as statements of aquatic life use. General aquatic life use, they can be just appallingly vague, truly appallingly vague, or they can be extremely sophisticated and well-defined and well-articulated. So these are just some examples so you have some idea. A couple of these examples are examples of tiered uses where there's allowances for quality that retains, setting goals that would retain an existing very high quality condition, and then other numeric criteria that would assess at a kind of lower expectations based on widespread disturbance. I'm not going to go into any detail about tiered uses, but just to point out that there's a great variety of ways that states have worked with the concept of aquatic life use. So in order to actually establish what these goals mean that are stated in water quality standards aquatic life uses, it really does require to do it well and intelligently. It requires a real marriage of science and policy. The public policy task is one that organizations such as the River Network partners and environmental advocacy groups can play an enormously important role on in advocating for really substantial progress in biological assessment science. But it comes down to a task that often falls to managers and legislators to weigh the socioeconomic costs and benefits and the environmental costs and benefits of how these thresholds are set. It certainly is best if it's a very transparent process. I would argue it's essential for it to be a transparent process, and right now there's a pretty significant lack of transparency in what these criteria thresholds actually represent in terms of condition, and then certainly also requires public participation. The technical task is really takes the skill of scientists who understand a lot about not only about ecology and aquatic natural functioning of aquatic systems and how stressors affect those, but also certainly it advances best when those scientists are applied, when they actually have an understanding of what some of the public policy aspects of the task are in order to set these biocriteria thresholds. These types of scientists are somewhat few and far between, but they've been referred to as policy entrepreneurs. They're scientists who understand the science, but also have a deep understanding of the policy context that science, for that science to actually do any good. And then local ecological knowledge of the biological potential and reference conditions is certainly essential. And credible bioassessment programs are what's required to actually make this work. So I have a very, I'm going to quickly run through just an example of how the policy and the technical context looks for an example from the state of Maine. So we begin with the same federal law to restore and maintain the physical, chemical, and we're focused here on the biological integrity objective. And then in Maine state law, this is in statute, we have established narrative biological criteria, which is a little different than the numeric that I've been talking about, but they have the same power of law, and it just depends on how well defined they are, how well a narrative biocriterion can be applied. But basically in Maine state law, we've specified quite precisely the biological condition goals for our various classes of aquatic life use. And in Maine, we established a tiered classification system so that we could protect our very highest quality waters at that high level with the forcing power of statute, of the narrative standards in statute. And then in statute also we have included these operational definitions that really interpret what the narrative standard means in a meaningful way that scientists can really understand what the legislative intent behind this language is. That's also in statute. And then our numeric biocriteria are in rule or regulation, where we actually specify the methods that are used to determine attainment or non-attainment. And in Maine, we use a statistical, fairly complex statistical model, a multivariate model. Again, very quickly, just as an example, this is an example of a tiered aquatic life uses. So very high quality class AA and A down to class C, with the narrative criteria listed over here as naturally occurs for AA and A, and then no detrimental changes to the biological community for B, and maintain structure and function for class C. In Maine, about 90% of our waters are managed in class AA, A, or B, and so a relatively small number of river miles are managed as class C. And then translating that, the narrative standard in statute here to what, as scientists, we thought could, the ecological value that was being communicated in this narrative standard. And this isn't spelled out. This is more of a logic train, I guess, that's depicted on this graphic, with the different sorts of attributes or aspects of the assemblage information that can be assessed that would pertain to determining whether, okay, do we still have a natural assemblage here, or is this unimpaired? And so that's what's being communicated in this central list. And then the quantifiable measures, these are actual metrics or ways of working with name and count information, organism name and who and how many. So So that's how it kind of translates in one state anyway from the law, the words in the law, to the actual number crunching and statistical analysis aspects. And then for the best programs that really have that tight connection to law, then that data can be used in all of this whole alphabet soup of Clean Water Act and water quality standards sorts of sections. So it becomes enormously important and valuable for really making a determination about how things, how an agency or a state or a region is actually performing relative to all the mandates in the Clean Water Act. So again, we come back to this this image of the collection of the environmental outcome information, and this is the biological aspect, going through a look at the list of impaired waters and how the TMDL, the information from the environment feeding into all of these things, modifying permits to get a better biological outcome. And then asking the question about, first you have to identify your highest quality waters and unique biological resources, figure out how to achieve the highest use in those, and improve degraded waters and so forth. So anyway, it's just this corrective process that ends up providing the agency with crucially important information about how well it's actually doing in achieving the results that they say they intend to achieve. So I have. Do you want to pause here again, Merritt? That was quite a lot through that section.
It was. So again, if people have questions, feel free to use the Submit Questions box, or if you're on the phone and you want to be verbal, you can hit *6 to unmute yourself and ask your question. Well, it looks like some people, oh, here, I've got one pop up. Okay, Mary says, can you explain, in quotes, a credible bioassessment program? I think quoting, the importance of such a program on one of your slides. Yeah,
a little bit later, I have a few slides in here on some mechanisms that have been set up to provide a peer review of biological assessment programs. It's kind of a quality assurance review called the Critical Technical Elements Evaluation. So there's a great deal that is available, or almost available. I'm not sure how publicly available it is, as in being up on an EPA website, but the Critical Technical Elements Evaluation is a process that has been applied to, I think at last count it might have been 23 or 24 different states around the country, that really is an exhaustive analysis of how proficient their program is and how well the stated intent of using the biological information is actually feeding into the management aspect. So that the question is asked about whether the biological information is actually able to be effectively used in action forcing or in internal agency, modification of practices in order to better optimize the outcome. So the short answer, I have trouble giving short answers, but it's basically the critical technical elements evaluation is a really important and good starting point for figuring out whether there is a credible program. And there are I've participated in those evaluations for the New England states. Chris Yoder and the Midwest Biodiversity Institute is really the lead contact, and Susan Jackson from Headquarters National Biocriteria Program would be able to let you know if your state has had a critical technical elements evaluation or not. It's a great process.
Mm-hmm. Does anybody else have any questions? I'll throw out one, Susan, just because we're doing well for time, and it's probably a little bit of a leading question on my part, but you mentioned in your earlier section that in some ways the science that we're talking about here has moved beyond where the states and EPA have yet gone in terms of the policy side of things. So the science is actually a little ahead of policy. Can you comment on why that might be? Is it just the resource challenge of developing some of these a credible program? Is there actual resistance to the idea? Is there any way you can characterize that disconnect a little bit for people?
Well, there are a lot of different tacks that I could take on how to respond to that. I think at its simplest explanation, there's just an enormous amount of inertia that there have. The Clean Water Act drivers early on, and appropriately, were focused on wastewater permits and controlling, really egregious chemical water quality pollution. Human health concerns equally egregious to aquatic life concerns, and so. For good or ill, enormous institutional structures grew up around that permitting mentality, and so, one again big picture way to look at it is I've heard Chris Yoder refer to is that most of the institutional structures that are in place now are really kind of sitting in the office looking. It's an enormous investment in wastewater discharge permits, which are tremendously important, and most of the, a huge amount of progress in water quality has been made through those kinds of engineering and permitting solutions. But there's little left over now for actually being in the resource and looking back up from the perspective of the assemblages that are being affected, and so there's kind of a mentality that if the permit, if you get the permit right, then you can kind of wash your hands of worrying about anything else. That should pretty much do it, and of course that's not the case at all. The combination of stressors that are having an influence on what can survive in the resource are enormously complicated and often go undetected. The impacts go undetected unless you actually look at the outcome. And so it's, limiting, shrinking resources. Much of it does also go back to a legacy of kind of what I've called the institutional persona. Many water management agencies in the states were started by engineers and chemists, and so then that becomes kind of the personality type. Right. Almost. Those that have a strong, started with some strong biological perspectives, maybe had an easier go of prioritizing that way of looking at things. So there's many answers to that question. It's important, though.
Okay, thanks. Any other questions before we move on? All right, well, if you unmuted yourself, please hit *6 again to mute yourself, and take it away, Susan.
All right. So there are many purposes for looking at the stream itself and the biota that are able to survive, but some of the uses that we have made of biological monitoring information is to basically just look at the ecological health of rivers, streams, and wetlands. This practice helps to identify if there are rare or exceptional aquatic assemblage resources there. For Maine, because we have a tiered system, we can look at a water body that may have kind of been by default put into a class B, which is a good intermediate class, but not our most protective class. And we can then come back and say, Well, it's assigned by the legislature to be managed at Class B, but it actually is a very high-quality resource, and it's biologically attaining Class A, and so then we can propose to reclassify that water body so that those higher conditions are maintained by reassigning it to a Class A management goal. So that would get into this second bullet, determine highest attainable biological goals or highest attaining biological goals. We have a lot of high-quality water in Maine. Most states, I would argue, also do have a lot of currently attaining high biological conditions, but they're not often managed in ways that will maintain those high-quality conditions. Of course, also a lot of waters are not attaining their goals. Urbanization is certainly very widespread impact even for a state like Maine and for other states. I just mentioned to Merritt, I've just recently come back from Belgium where I was participating in some it's basically the European Clean Water Act work, and the just really astonishing, age of the cities over there in Europe. I was in Brussels, and they're, very different expectations for what really is attainable there, with so much impervious cover and such long-standing and widespread disturbances to natural habitats. Biological assessment also is very helpful in defining what's causing the problem. This is something that takes a lot of research and development, again, that has not been fully exploited. The science that's out there has not been fully exploited by states, but enormously helpful in development of total maximum daily loads that actually can help to move the biological condition to a higher quality. Or to the attaining quality, and then to evaluate the overall effectiveness of management actions. Great. It's just a great experience. To be a field biologist is a wonderful way to spend your time. These images are, this is Leon, and he's testing the flow here. This is Tom with his head in the periphyton bucket, and he's counting the algae cover in this quite lovely river here. And this is John, and he does fish tissue sampling, so they collect a great deal of fish throughout the summer and analyze them for fish tissue contamination. The method, I could show dozens and dozens of different sampling techniques. My readily available slides have to do with our methods that we use in Maine, but we use these artificial substrates that are placed in the river bottom for about a month to 6 weeks, depending on the type of habitat. And then we collect all the organisms that colonize these artificial substrate samplers. These are a little closer look. These are placed just by weighting. We also have a method in Maine, and some other places use this, but not too many, where we can sample in non-weightable rivers. So this would be placed by a boat down. It would just drop a line down, and this rests on the river bottom, and then it's got a screen on the bottom of the sampling device so the organisms aren't lost. And this little Wizard of Oz hat here goes down and closes over the sampling device to retrieve an enclosed sample. And there, just again, some images from the field with cleaning off the organisms from the rocks, and then you get you get whatever you get, and then that's all worked up in the lab and analyzed, statistically analyzed. We touched on some of the points made in this slide. This is perhaps a little harsh, but I don't think it's untrue. often the technical programs have been sort of a poor stepchild to the larger, more chemically focused programs within management agencies. The result, when you don't really have a strong connection between the biomonitoring data that may be brought in. Just about every state collects lots of biological monitoring data, but biocriteria or in really carefully defined aquatic life uses in water quality standards, then it's very difficult to actually bring a case or influence things. And this is part of one of the aspects that the critical elements evaluation looks at. So biological declines across the country, there have been some summaries of this, but they don't really very frequently trigger management intervention, and this has been demonstrated unless there's a kind of an egregious change. Now that's a blanket generalized statement that certainly isn't the case everywhere and for every instance, even in a program that doesn't have very strong standards. Often things can be brought to bear to change a bad situation, but by and large, we could do better. Goals are poorly expressed and poorly addressed, and that's a water quality standards tiered aquatic life use description problem. And we've talked a bit about these outdated policy structures. Pollutant by pollutant TMDLs, this was totally appropriate when the concept of TMDLs were first put forward, and in many cases is still totally appropriate. If there's some industry that's just discharging too much chromium, then it's a really good idea to have a pollutant by pollutant TMDL. However, there are many other situations now, for example, with urbanization, where that makes no sense whatsoever. It's very frustrating and difficult for these suites of land use alteration disturbances to be restricted to the outdated language about TMDL, so that it makes it very difficult to actually correct the problem. And then just the lack of resources to prioritize implementation of biocriteria, program development on the technical side, and use of this is an EPA procedure, stressor identification, that's really very well thought out, but it's a quite exhaustive process that has, some states are able to pick up on it, but it's mostly at their own initiative. Do we want to pause again here a bit, you think?
Why don't we quickly? I don't have any pending questions, but if anyone has a question, again, the chat box there is your one option. And if you're on the phone, you can do *6 in order to unmute and ask your question. It does look like we have somebody typing, so maybe we'll just wait for a second here, Susan, and see if that question pops up. If it doesn't, we'll hold it till the next stopping point. Depends on how long it is, I guess. Oh, here we go. The question is from Greg, and he says, does Maine or other states vary land use policy based on the quality of the water body?
Let's see. Well, yeah, I guess the way I'll interpret that question is we, in Maine, well, we now have a different political climate I won't go into, but historically we have had pretty much statewide land alteration laws and rules, Shoreland Zoning and Natural Resources Protection Act. These are state laws that generally apply statewide. I know many other states will have a much more local or small regional, maybe county control over land use permitting and land-based development activities. But nevertheless, in Maine, where we do have a tiered classification system, some of the provisions for permitting in a very high quality Class A water body can be applied differently, or will be different, or there may be enforcement actions brought differently than for a Class C water body. So the answer to that, does Maine or other states vary land use policy based on the quality of the water body? I think for states with tiered aquatic life uses in place, that's a fairly straightforward answer to that would be yes. For other states, the kind of the wishful thinking, in my opinion, answer is that well, anti-degradation will hold the line there, but it's very experimental in terms of applying anti-degradation to biological alterations, biological changes that are, maybe, it's pretty much you have to predict what might happen, and so it's anti-degradation. I think is quite ineffective. It was really designed for little increments of increasing chemical pollutants in a permit, and how much. Anyway, I won't go into anti-degradation other than that. In theory, it should be great, but I really don't feel as though it works very well for biological changes. So many states, some of what we were looking at in examining how states are performing with their how much muscle their biological assessment work has. We found that there was really very little they could do, even if they knew it was a high-quality condition. There was very little that they could do with the power of their program to intervene.
Any other questions from anyone? All right, we don't have any more on chat, Susan, so you are free to go ahead. If you're on the phone, make sure you've muted yourself, and go on ahead to your last section here, Susan.
Okay. So here, again, this slide is a little bit scrambled, but basically this y-axis here is biological condition from very high quality up here at the top to a low quality down here, severe changes. What this slide is showing is across the top here, levels of proficiency of bioassessment programs, and this is all spelled out, level 1, 2, 3, and 4 in the critical elements evaluation that I mentioned earlier. So a level 1 program is basically a pass-fail kind of a very broad, oh yeah, it looks really good, oh well, it looks really bad, but we're not sure what's going on in the middle. A level 2 program can tighten it up a little bit better than that, but still a very coarse assessment, kind of a one-size-fits-all. You either pass or you fail. Unfortunately, if the program has such poor technical proficiency that it falls into a level 1 or a level 2, usually the concern is not to overreach and say that it's failed up here at this level. They'll wait until it gets all the way down here so that there's no legal jeopardy that the agency is saying that it's failed. So there's a great deal of difference between a pass and a fail, and the fail bar is often set at the lower end of whatever the threshold is that's been established. So anyway, then a level 3 program is getting more and more refined, able to distinguish, and then a level 4 program, there's only 3 level 4 programs in the country right now, but they're able to really refine thresholds, through many different types of changes.
Susan, do you know off the top of your head the 3 level 4 programs? People might be interested to know where to look.
Yep, Ohio, Maine, and Vermont. Connecticut is in a very rapidly improving, technically improving mode right now, and they they may be pressing up against level 4, and Minnesota is also making a very serious investment in their technical program. But these assessments, the program evaluations are done at a point in time, and so they would need to be reevaluated to see exactly where they came out. But that's where things stand right now.
Great, thanks.
Okay. Strong technical programs is very, very important. This is, again, the slide looks a little bit scrambled, but basically just a kind of a wave it in front of you to show the types of things that are considered. And then there's an elaborate scoring system to, it looks like the slide is also kind of being cut off a bit, at least on my screen. But you don't really need to know the details other than to know that a 200-page document exists that goes into exhaustive detail about each of these elements and why they're important and how the scoring is established. So these are what go into the evaluation of program proficiency. The other concepts, you know, I think I'm going to use this little feature up here with the guy raising his hand. If people could say whether or not you have heard of the biological condition gradient, I would be very interested to know it. And you can drop down that little I don't know, Merritt, maybe you can explain it better. But I'd love to see people raise their hands if they have heard of the biological condition gradient.
Susan's talking about the button she mentioned at the beginning of the presentation. On the black bar at the top of your screen, there's an icon with a little man raising his hand. If you click on the arrow next to him that points downward, it gives you options. So select raise hand if you have heard of the biological condition gradient.
Oh, cool. I can see some of them.
Can you see?
Yeah, oh, that's very nice.
It looks like we have a chunk, maybe almost a third, approaching a third of the people have heard of it. Okay.
Well, that's great. So I think that this presentation will be at about the right level of detail with it. It's basically an EPA and state collaborative initiative that I had the privilege and the really stimulating opportunity to participate in. I did a year down at EPA headquarters at the National Biocriteria program, and this biological condition gradient was an outgrowth of that time working with Susan Jackson down there. But it's basically a descriptive model that partitions. I'll go into it in more detail in a couple of other slides. But it's looking at how biological condition declines across increasing disturbance and stressors, and it provides a great deal of descriptive detail about what these different condition steps will be as you're declining down this gradient of increasing stress. The really useful thing is when the water quality standards context can be directly related to the science context, as I mentioned earlier. So this example from Maine, a tiered water quality standards framework would directly articulate or map the goals stated in tiered aquatic life uses to where that would fall on this biological condition gradient. So that's just a quick little kind of an icon description of why the biological condition gradient is useful and important. I'm going to just get into a little bit of detail about it. Observations from many biologists across the country, and we do see these patterns of predictable change. Let's see. So this is the slide that doesn't work for whatever reason. There's supposed to be a little table in here, but I can still kind of illustrate. The biocondition gradient has these six different levels. This would be a tier 1 up here, very good, intact, beautiful, intact watershed, and this is the type of critters you would expect in a naturally derived place. Down here, this is that stream draining a shopping mall, and it's all turned to tolerant organisms. So the biocondition gradient provides a whole bunch of detail about how those different steps change, and then the intermediate step in the middle there. It's a conceptually complete model. It is a conceptual model, but it has in many places been elaborated into a calibration of actual in-hand data, empirical data that's collected from the resource. But the model describes condition from natural to severely degraded in these 6 steps. Grounded in first principles of ecology. It was very well vetted through academic and applied scientists. And it's very, very helpful in organizing and simplifying extremely complex ecological data. The thing that's important about it for organizations like River Network or environmental advocacy groups is that it can, if you just even grasp the very most basic aspects of it, it can add a great deal of transparency to communication of environmental results information. This is again sort of an iconic depiction of it. I'm not going to go into the detail about the science of it, but it's got these little thumbnail descriptors here that fit onto this type of a depiction of the biocondition gradient. But within each one of these tiers, the tier 3, tier 2, tier 1, are 10 ecological attributes that are described in great detail as they change as you go down the progression, getting to worse and worse conditions. It can be really important in terms of reference conditions. This series of slides here depicts an area of the country that may have widespread disturbance and very little that's really remaining to derive biocriteria from, and that would mean your best conditions that are still out there would be right here, which is way down it might be at a tier 4 or 5. This is a pretty extreme example here, but maybe in Brussels, I bet they're easily down here. So you would be saying departure from this point here when you're trying to show your bioassessment, your biocriteria would be the best we have is right here, and this place is over here, so it's that bad. When in fact in a pristine situation, the best you have is way up here, and you might find another disturbance regime that would be at this level. But you would be able to have this full gradient to really describe that departure from reference. So this is a kind of a crucially important concept in bioassessment and biocriteria development, to know where your reference conditions are and to hold the developers' feet to the fire about really being clear and articulating what reference conditions are being used to set the biocriteria. Another nice thing about the biological condition gradient is that all of the states, they used to call it at EPA letting 100 flowers bloom, and so each state has kind of gone to its own approach to develop their numeric biocriteria to the extent that the states have biocriteria. But through the biocondition gradient, those should all be able to be related back to, if you have an outcome of some percentage of observed over expected, say in this RIVPAX model for Oregon, some particular percent outcome should represent something on the biocondition gradient that is also represented by some Maine outcome that would also represent a tier 3 or a Florida one, even though they're using completely different methods. As I mentioned, I had this terrific opportunity to participate in a peer review of the European Water Framework Directive. I just got back yesterday from this trip to Brussels, and the European Water Framework Directive is using a concept very, very parallel to the biocondition gradient. In fact, their descriptors are not as detailed as the biocondition gradient, and they have five chunks or 5 categories rather than the 6 in the BCG, but a very parallel sort of a framework. Quite interesting experience over there. Another depiction of the biocondition gradient, just to illustrate that in actual calibration of it or development in different water body types using the actual data that it's taking place for wetlands. There's a pretty well-established wetlands biocondition gradient conceptual model, an algal, Maine algal biocondition gradient that's very detailed and has been fully calibrated to numeric biocriteria for algae, and then a large river fish biocondition gradient for large rivers in the Northeast. There's also an estuarine biocondition gradient model that's being developed by EPA and some of the National Estuary Program partners. I think we're coming pretty much to the end, finally. This slide will give you access to a pretty good website. It's getting a little bit outdated, but it does have some good resources here with links to publications. Actually, the journal article on the biocondition gradient is up here. Unfortunately, the critical elements, I think you still have to contact Susan Jackson or Chris Yoder directly for that, because EPA has not released that document yet. They tend to develop drafts of certain things that make them, I don't know why, but anyway, it's just not out there yet. It's a very well vetted process, and it is available if you contact her directly.
And Susan, I would add to that, I have found if you talk to your state agency staff involved in biocriteria or bioassessment, they usually know if such an assessment, if it was done, and if it was done, they usually will share the results.
Oh, that's a great point. Yep, yeah, that's right. Certainly the communicating with the state staff is a great idea, and there is a network of folks who meet at regional and national meetings and talk about this sort of thing with their EPA regional contacts and so forth. So that's a great suggestion. And I guess that's it. So just a few minutes left here.
Yeah, well, perfect timing, Susan. Let's see if there's any questions about anything we covered or anything you were hoping would be covered that we didn't quite hit on. This would be a great time to pipe up. We have a few minutes here for questions for Susan. Again, *6 yourself to unmute if you're on the phone, or go ahead and type your question in. I can see there's several people typing, so while they're doing that, I will remind people that when we wrap up here, as you log out of the webinar, an evaluation form will pop up, and if you can take a minute to fill that in, it'll be really helpful to our presenter and to us for future webinars. So please do take a second to do that if you can. And again, it looks like, okay, here we got the first question, and it looks like at least one more is on its way. So Judy asks, will a copy of Susan's slides be available for download? What we do, and if this does not suit your needs, please let me know, and we can follow up individually. There'll be a follow-up email that goes out after this webinar. It'll actually take a little bit of time because we have recorded this webinar. We'll get that whole recording online and then send out a link to the recording that you can use if you want to go through it again, or if you want to share it with other people you work with, etc. So it will be available as a recording, and we'll also go ahead and include in that email some of those links that Susan put up, because some of them were long and hard to copy down. So we'll provide those in the email. Again, though, if you need copies of the slides as a PowerPoint or something specific, please contact me or Susan, and we'll see what we can do. It's a pretty big file. Barb asks, role, value, and use of BA, I like the BM, I guess biomonitoring, bioassessment, and biocriteria outside of Clean Water Act policy. Oh, that's an interesting question. Can you comment on that at all, Susan? In Maine, has it been used outside of the Clean Water Act world, or have you heard of it elsewhere?
Well, certainly there are watershed groups, and I'm trying to think. I guess I know that there are some municipalities that I'm thinking of in Texas that have, developed bioassessment programs in some detail. Those are still in the water quality standards context. But it's certainly been very valuable in educational roles, and some places that have developed citizen monitoring initiatives have brought problems to the attention of the agencies, or just gotten public interest alerted based on findings from more citizen monitoring. I guess I would be interested if you had, Barb, some thoughts or more specific questions about what you're seeing. If your point is what can citizen groups do in terms of monitoring, or if it's I guess the other role for bioassessment and biomonitoring has been with other federal programs. USGS has enormous, the whole NAQUA, National Water Quality Assessment Program, used tremendous bioassessment efforts, as well as physical, chemical, hydrological, and assessments to look at water quality throughout the country over the last 10 or 15 years. We were involved in a process where they used looking at urbanization gradients in different parts of the country. There's quite a bit online. Effects of urbanization on ecological systems, EUSE, if you're interested in that. And those partners did some terrific work looking at biological impacts of urbanization, and actually developed an interesting model that relates it to biocondition gradient tiers. So we worked with them up in the Northeast a bit. So those are things off the top of my head. Certainly a lot of folks academically also go and look at biomonitoring type research projects to try to identify causes of observed biological impairments, and trying to link stressors to observed biological impacts. That work is always ongoing.
Any other questions here before we are going to hit right on time here, Susan? Thank you so